Hi, my name is Joel Nitta. Thanks for coming to my talk today. You can also view these slides as a web page from this link. Today I will discuss our recent work, Exploring Dimensions of Biodiversity in Japanese Ferns. Understanding the distribution of biodiversity is critical for conservation in the face of anthropogenic change. The vast majority of studies analyze biodiversity by counting species. However, we know that species are really just the tips of the tree of life. To truly understand biodiversity, we should also take into account other dimensions, such as phylogenetic diversity, functional diversity, and their interactions. This is not an easy task. The main reason few studies have done so is because it requires immense amounts of data. Fortunately, there are some systems that are amenable to such an analysis. Here, we introduce the ferns of Japan as a model system for studying biodiversity. They are quite diverse overall, including nearly 675 species representing 98 genera and 34 families. They are extremely well sampled. In fact, according to GBIF, they have by far the most collections of any country. Nearly complete sampling is available for DNA, distributions, and traits. Japan is an extremely interesting area to study biodiversity because of its biogeographical history. It spans a wide latitudinal band going from subarctic climate in the north to subtropical in the south. This, coupled with high variation in elevation, has resulted in a remarkably rich environmental diversity over a relatively small area. Furthermore, periodic connection with the mainland has allowed Japan to serve as a refuge during interglacial cycles and influence diversification in the region. The goals of this study are threefold. Using the ferns of Japan as a study system, we seek to understand: how is biodiversity distributed? How is it structured? And how well is it protected? First, I'll explain our data sources and then describe methods along with the results. Our occurrence data come from a list of specimens at herbaria in Japan, including about 300,000 specimens representing 764 taxa. We converted this list to a 0.2 degree presence-absence matrix to analyze diversity patterns. The molecular data include RBCL sequences representing 97% of the taxa. However, RBCL alone is insufficient to generate a completely resolved tree, so we combine these sequences with a much broader sampling from GenBank, use this for phylogenetic analysis and dating, and then finally trim the tree back to only the Japanese taxa. Our trait data include 16 morphological traits that were originally used for species identification. Now for the first question: how is biodiversity distributed? Species richness shows a latitudinal hump-shaped pattern reaching maximum values on the island of Kyushu. This is a well-known pattern, and it is probably related to favorable climate for ferns combined with steep elevational gradients. So that's species richness. What about the other, less studied aspects of biodiversity? We measured phylogenetic diversity, or PD, with face PD, which is the total branch length summed across all species in a community, and functional diversity, or FD, using face PD on a trait dendrogram. So it turns out that the raw values of both PD and FD are highly correlated with taxonomic richness. As we can see, all of these heat maps look nearly identical. Now, this may seem a bit underwhelming at first, but this is actually the expected result. As you can see from these scatter plots, PD and FD increase rapidly with richness and then start to level off. This is expected because the more species that occur at a site, the greater is the chance that they will come from different parts of the phylogeny. What is more interesting is whether the observed values of PD and FD are more or less diverse than expected by chance. And we look for that using standard effect sizes. The standard effect size, or SES, is the difference between the observed value and the mean of a null distribution divided by the standard deviation of the null distribution. SES shows how extreme the observed value is relative to null expectation. So in our case, 0 is no different from random, positive values are more diverse, and negative values are less diverse than expected. So let's look at the SES values of phylogenetic diversity. Here we see that SES of PD is generally low throughout the region. This pattern is often observed at spatial phylogenetic analyses at broad scales, because the study area encompasses multiple biomes. Species niches tend to be conserved at this deep level, such that no site includes representatives of all the major lineages. Another way to analyze phylogenetic diversity is to look at relative branch lengths. Are branches on average longer or shorter than expected for a given community? We do this by comparing PD measured on the original tree to PD measured on a tree where all branch lengths have been set equal. This metric is called relative phylogenetic diversity, or RPD. High RPD indicates long branches, which could be due to species evolving in isolated refugia, or to immigration by species that are distantly related to others in the community. Low RPD indicates short branches, which could be due to recent speciation. The ferns of Japan show high RPD at both high and low latitudes. In the north, this is likely due to certain north temperate taxa on long branches, such as Equisetum and Batricium. In the south, it is probably due to many tropical lineages that are much more diverse outside of Japan, but have highly restricted ranges within Japan. In fact, 4 families occur only in the southern area and not on the main islands. Conversely, the low RPD of the central area may be due to clades that have radiated on the main islands, such as Dryopteris. We can also look at the functional analog of RPD, our FD. We see a somewhat similar pattern, with high diversity in the southern islands and low diversity elsewhere. Again, this is likely due to tropical lineages with unique morphologies. The last component of biodiversity we'll look at is endemism. Phylogenetic endemism, or PE, is phylogenetic diversity inversely weighted by range size. So, communities with small ranging taxa that occupy unique parts of the tree will score highly. We see once again that the southern islands are by far the highest for PE. This is almost certainly due to the fact that these southern subtropical islands are very small in area compared to the rest of the country. So, the take-home message from this section is that although they are not the most diverse in terms of taxonomic richness, the southern islands are highly endemic and diverse in terms of their phylogeny and traits. Let's turn to the second question. How is biodiversity structured? We analyze the structure of biodiversity using the R package phyloregion. In short, this involves measuring beta diversity, or the differences between sites, with either taxonomic or phylogenetic metrics. We then use an algorithm to group these distances into a certain number of clusters and determine which number of clusters fits the data the best. This process resulted in 8 taxonomic bioregions, but we can see that the vast majority of sites fall into just 4. Region 1, in purple, includes Hokkaido and the northern part of Honshu. Region 2, in green, corresponds to the southern half of Honshu, Shikoku, and Kyushu. Region 3, in blue, corresponds to the islands south of Kyushu, and Region 4, in yellow, corresponds to the Bonin Islands. Each of the major bioregions are well represented by certain species. So next I'll show some examples of species distributions that exemplify each region. In Region 1, we have species of Diperia and Dryopteris. In Region 2 are species including Dryopteris hikonensis, Diplopterygium glaucum, and Poliscatum palablepharum. In Region 3, the southern islands, we have some Pteris and Nephrolepis baserata. And Region 4, the Bonin Islands, include some endemic taxa. Okay, so what about bioregions analyzed using phylogeny? Well, it turns out that the phylogenetic bioregions are actually quite similar to the taxonomic bioregions. This suggests that bioregions in Japanese ferns are due to both tip-level diversification as well as deeper splits in the tree. Interestingly, the bioregions identified here with ferns match those traditionally defined on an ad hoc basis for forest types. This suggests that ferns can be used as good environmental indicator organisms in Japan. Finally, we'd like to know how well protected are the biodiversity hotspots that we've identified here. One thing that is important to understand first is that Japan is very well protected overall. Here we've classified protected areas into 3 categories based on the level of protection. Approximately 30% of the land area in Japan falls into one of these categories. So here I'm showing hotspots of species richness, that is, the sites with the top 5% of species richness. They're all located in central Japan, and as you can see, all of the sites have at least some level of protection, including a few with the highest level, so that's good news. Now turning to endemism, these are the sites that have significant PE compared to the null distribution, and they're all located in the southern oceanic islands. There are a few sites that are not protected, but overall, again, the protection level is quite good. Unfortunately though, the story of fern conservation doesn't end here. it turns out that there is another major threat to ferns in Japan, and that is the native shika deer, Cervus nippon. When populations of these deer are dense, they can really decimate the herb layer, as we can see in these photos. Now, populations of this deer have grown exponentially since the 1970s due to decrease of natural predators and hunters and climate change. Of course, deer don't respect conservation zones, so as you can see on the map on the right, there is evidence of deer damage even in national parks. There is an ongoing effort to reduce deer populations and protect particularly fragile plant communities with exclusion fences, but the problem is far from resolved. So, in conclusion, I wanted to know three things about the ferns of Japan. How is biodiversity distributed? How is it structured? And how well is it protected? We see different aspects of biodiversity are distributed differently, with taxonomic diversity highest in the main islands on Kyushu, but phylogenetic and functional diversity highest in the subtropical southern islands. The ferns of Japan are mostly structured into four main groups that track traditional forest types. They are well protected in terms of their conservation status, but they are in danger of herbivory by deer. So there are many more topics to further investigate using this remarkable study system. One is to understand the role that hybridization has on diversification. The ferns of Japan also include nearly 300 described hybrids, but I did not include those at all in this analysis. Additionally, I would like to know more about the role of gametophytes in community assembly at broad scales. So with that, I'd like to thank the organizations and individuals that made this research possible, and all of you for listening to my talk. Thank you very much.